Hello students, this is the 14th video of molecular basis of inheritance and topic of today is translation. So let's start. So what is translation? Translation is formation of proteins from mRNA. So if mRNA forms proteins, it is known as translation. So formation of proteins from mRNA is translation. The site of translation is ribosomes. Because ribosomes are site of translation, so they are also called protein factories. Now let us look at steps of translation. There are three steps in translation. The first step is charging of tRNA. The second step is initiation. The third step is elongation. And the fourth step is termination. Now we will look step by step at these steps. So let us look at the first step, that is charging of tRNA. Charging of tRNA is also called amino acylation of tRNA. Now, in the charging of tRNA, the tRNA, that is transfer RNA, is linked to their respective amino acid in presence of ATP. So what will happen in charging? The tRNA would be linked to the respective amino acid in the presence of ATP. And the enzyme required for this process is amino acyl tRNA synthetase. So it is also known as amino acylation of tRNA. Now let us understand this step. Suppose this is the enzyme, that is amino acyl tRNA synthetase. So this enzyme will have active sites. In the active site, there would be ATP, there would be tRNA, and there would be methionine. So in the presence of this enzyme and in the presence of ATP, the tRNA, which is shown in pink color, would be linked to the amino acid. Here the amino acid is methionine, and this step, since the tRNA is linked to the respective amino acid, which is methionine here, is known as charging of tRNA. Now the next step after charging of tRNA is initiation. Now during initiation, the mRNA will bind to smaller subunit of ribosomes. Now let us understand this with the help of diagram. Now as you can see in the diagram, mRNA has binded to the smaller subunit of ribosome. So when mRNA binds to smaller subunit of ribosome, this is the first step in initiation. Now, when the smaller subunit of ribosome binds to the mRNA, in the next step, the larger subunit of ribosome will bind to the smaller subunit. As you can see here, this which is shown in red color is the larger subunit of ribosome, and it has binded to the smaller subunit of ribosome, which is already binded to mRNA. Now here one must remember that the larger subunit of ribosome has three sites, that are E site, P site and A site. E site is called exit site, P site is called peptidal site, and A site is known as amino acyl site. Again I am repeating, larger subunit has three sites, E, P and A. So first smaller subunit binds to the mRNA, smaller subunit of ribosome. Then the smaller subunit binds to the larger subunit. And the larger subunit has three sites, E, P and A. Now the whole process of initiation requires an initiation factor. So until now, smaller subunit has binded to larger subunit, and the larger subunit has three sites, E, P and A. Now on this P site would be present a initiation codon, that is AUG, And now the tRNA with its corresponding amino acid, because the tRNA is already charged, will bind first to the P site. One should always remember that during initiation, the amino acid and the tRNA complex will bind to the P site, that is peptidyl site first, which is present on the larger subunit. So as you can see in the diagram, at the P site, methionine tRNA complex, or we can say amino acid tRNA complex, has binded. Now, the third step of translation is elongation. So what will happen in elongation? We know one tRNA which has methionine with it has already binded to the P site. Now another tRNA will come and it will bind to the A site. Suppose that the tRNA which has binded now to the A site carries amino acid 1. So we will call it tRNA having amino acid 1, which will bind towards the A site. So now we have two tRNA. One has binded towards the P site and another has binded towards the A site. Now between these amino acid, that is methionine and amino acid 1, a peptide bond would be formed. And this bond is formed with the help of enzyme peptidyl transferase. Now, when the peptidyl bond is formed between methionine and amino acid 1, what will happen is that the tRNA present on the P site will move towards the E site, and the tRNA carrying amino acid 1 present on A site will move toward the P site. So the P site tRNA will move toward the E site and A site tRNA will move toward the P site. That means shifting of tRNA has occurred, and this is known as translocation. Now let us understand this with the help of a diagram. As you can see here, the tRNA which was carrying methionine has moved towards the E site, and now it has exited from the ribosome. It has moved away from the ribosome, and the tRNA carrying amino acid 1 has moved toward the P site. But remember that when the tRNA shifts and moves away from the E site, the amino acid which it was carrying, that is methionine, will remain attached to the amino acid 1 because there was peptidyl bond which was already formed. Now another tRNA will enter the site A. Suppose it has amino acid 2 attached to it. Now what will happen now? The tRNA which is on the P site will enter E site, and again it will exit from the ribosome, and the tRNA which is on the A site will move towards the P site. But now it will have 3 amino acids. So as a result, we know the chain of amino acids are known as proteins. So the protein chain would be formed. Similarly, the amino acid will keep coming and be translocated from one side to another, and this chain of amino acid will elongate. Now, up to what extent will the amino acid chain will elongate? Now, this depends on the last step. The last step is known as termination. When the tRNA will reach towards the termination codon, that are UAA, UAG and UGA, the formation of peptidyl chain will stop and the translation would end. And we know that the chain of amino acid are known as proteins. So this is known as translation. When mRNA will form protein, this is known as translation. So if you have gained something from this video, please subscribe now to my channel Ramji Chopra, comment, like and share.